Some alleles are neither dominant nor recessive. In the crosses reported by Mendel, one member of each gene pair was clearly dominant to the other. Such behavior, however, is not universal. Sometimes the heterozygous phenotype is intermediate between the two homozygous phenotypes. For example, the cross between a pure-breeding red snapdragon, Antirrhinum, and a pure-breeding white variety gives F progeny of the intermediate pink colour. If these F1 progenies are crossed among themselves, the resulting F2 progenies contain red, pink and white flowers in the proportion of 1:1, figure 1:2. Thus it is possible here to distinguish heterozygotes from homozygotes by their phenotype. We also see that Mendel's laws do not depend on whether one allele of a gene pair is dominant over the other. Principle of independent assortment. Mendel extended his breeding experiments to peas differing by more than one characteristic. As before, he started with 2 strains of peas, each of which bred pure when mated with itself. One of the strains had round yellow seeds, the other wrinkled green seeds. Since round and yellow are dominant over wrinkled and green, the entire Phi generation produced round yellow seeds. The F generation was then crossed within itself to produce several F progeny, which were examined for seed appearance, phenotype. In addition to the 2 original phenotypes, round yellow, wrinkled green, 2 new types, recombinants, emerged, wrinkled yellow and round green. Again, Mendel found he could interpret the results by the postulate of genes if he assumed that each gene pair was independently transmitted to the gamete during sex cell formation. This interpretation is shown in figure 1:3. Any one gamete contains only one type of allele from each gene pair. Thus, the gametes produced by an F, RRY, will have the composition RY, 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 or RY, but never RR, Y, Y, or RR. Furthermore, in this example, all 4 possible gametes are produced with equal frequency. There is no tendency for genes arising from one parent to stay together. As a result, the FC progeny phenotypes appear in the ratio 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green as depicted in the Punnett square, named after the British mathematician who introduced it, in the lower part of figure 1:3. This principle of independent assortment is frequently called Mendel's second law, Chromosomal Theory of Heredity. A principal reason for the original failure to appreciate Mendel's discovery was the absence of firm facts about the behavior of chromosomes during meiosis and mitosis. This knowledge was available, however, when Mendel's laws were confirmed in 1900 and were seized upon in 1903 by American biologist Walter S. Sutton. In his classic paper, The Chromosomes in Heredity, Sutton emphasized the importance of the fact that the diploid chromosome group consists of 2 morphologically similar sets, and that during meiosis every gamete receives only 1 chromosome of each homologous pair. He then used this fact to explain Mendel's results by assuming that genes are parts of the chromosome. He postulated that the yellow and green seed genes are carried on a certain pair of chromosomes, and that the round and wrinkled seed genes are carried on a different pair. This hypothesis immediately explains the experimentally observed 9:3:3:1 segregation ratio. Although Sutton's paper did not prove the chromosomal theory of heredity, it was immensely important, for it brought together for the first time the independent disciplines of genetics, the study of breeding experiments, and cytology, the study of cell structure,